all. Good afternoon to all of you. I am Anirban Sengupta from University of Calcutta, India. So today I want to present my work which is entitled as the SQSTM1 P62. It is one of the key protein which actually has been found to play a key role in balancing the two cell death mechanisms, let's say type 1 cell death apoptosis and type 2 cell death autophagy, and also helps in the regulation of the antioxidant pathway, that is NRF2 keep 1 pathway in cerebral malarial infection. So we all know that malaria is one of the deadly diseases in the world, which claims a huge number of life every year. And from the WHO report, we find that almost 5 deaths every 4 minutes, almost over 1 death per minute. So by the time I complete my talk, by the time I completed my talk, almost within 10 minutes or 15 minutes, there are 10 to 15 more deaths there will be by this disease. So from this we find that this is one of the most killer diseases, and we also find that from that there is a huge cytokine imbalance during this mechanism of this malaria. So pro-inflammatory and anti-inflammatory cytokine balancing plays a key role, and we have already reported from our lab from the previous publications that how it helps or causes the killing of in the patients to die. So from that we find that there is one particular cell death mechanism, that is the autophagy. This plays a key role in controlling the infection. So the autophagy, as you can see from here, that as you can from here, that the autophagy has functions in adaptive immunity, the innate immunity, and also in the effector functions during infections like pathogen degradation, phagosome maturations, and also helps in the antigen presentations, so that the pathogens, once it's inside the cells, it can be degraded, and the autophagy plays key role in the degradation of the pathogen in many of the diseases. So, but it has not been reported about the autophagic mechanism in the host in case of malaria till death. So for that reason, we have preliminary stained our splenocytes. We have taken the spleen as the organ of the choice, because the spleen is the lymphoid organ, and the splenomegaly is important found to be important during the infection. So when we have done the formalin fixed paraffin embedded sectioning of the spleen and stained them with the acridine orange, which basically stains the low pH vacuoles. We have stained the MTC. Previously it has been the definitive marker for the autophagic vacuoles, but at the present time it has not been so. So we have stained the slide by the LC3B, a key autophagic marker protein for the immunofluorescence, and also done the transmission electron microscopy to see the double membrane vacuoles over here. We find that we have taken the control spleen and the spleen sections from spleens isolated, and that is harvested from the 4 days post-infection and 8 days post-infection mice. So from that we have also isolated the RNA and the protein, and from that we can understand the gene expression status of the autophagic marker genes, like we see the mTOR, LC3B, Backlin, P62, etc. in comparison to the GAPTH control. We have done the Western blot or the immunoblot analysis also to find out the expression of the few of the important proteins like P62, LC3AB, etc. And we find that all the autophagic marker genes are upregulated. Interestingly, to note that this P62 is one of the genes which need to be downregulated during the autophagy, because it also will get degraded as autophagy progresses. But we find some different results which contradicts with our other protein expression data. So we are interested to see that what might be the reason for this. So for that we are coming to that, but before that I want to show you one of the previous paper from our lab, so which shows that the splenocyte apoptosis in case of the Plasmodium infection. This is the Plasmodium bargei strain that we have used. This is the mice homologue of Plasmodium falciparum. So the Plasmodium falciparum, which is the killer pathogen, will not be able to cause the disease in mice. That's why we have to use the mice homologue, the Plasmodium bargei, and we find that the splenocyte apoptosis has been enhanced during this malarial infection. So now this is the autophagic flux. This autophagy is not a one-step process. This is a multi-step process, and we find that we want to see that whether the autophagy is blocked due to in one or the multiple in one step or the other. So whether the whole flux has been completed perfectly or not. Those autophagic marker genes show that there is accumulation of the initiation of the autophagy, but we are not sure about its completion. So that's why we have used the cationic amphiphilic tracer dye. This is from the Enzo. So this dye basically perfectly understands that which is the autophagic vacuole and stains that autophagic vacuole, and by that we can identify that whether there is an increase in the autophagic vacuole formation or not. We find the increase in the autophagic vacuole formation indeed. So with this we conclude that the autophagosome formation has been completed. Now we are interested to see that autophagy doesn't occur if the lysosome, that one of the important cell organelle, that lysosome, unless and until this lysosome fuses with the phagosome and form the phagolysosome, that is the autophagolysosome. We want to see that whether the autophagolysosome formation has been completed or not, and we find so that we stained our vacuoles with LC3B, only the autophagosomes, and lysosome by the lysotracker red, and if they are merged, then the signal should be red and green mix, that is yellow colored signal, and this yellow colored signal increases in the 8 days post infection. Hence we can conclude that the lysosome fusion is also occurring. We transfected our cells with the clone of GFP, RFP, LC3B. This is the double fusion protein with the LC3B, and if this LC3B protein is insert the autophagic vacuoles, the GFP is being the pH sensitive, and when the lysosome got fused, the GFP will degrade fast, leaving only the red signaling within the cells. So in our signal, we find that in our cases, we find that in 048 days post-infection splenocytes, we find that when they are transfected with the GFP, RFP, LC3B clone and superimposed with the Plasmodium, we find that that there is no such change in the signaling. That is, this green color signal in 8 days post-infection should be almost be diminished, because the green should be degraded if the autophagic vacuole, autophagy completion is there. So we have in doubt over here, and that might be a result, or that might be a cause, that the P62 that we find before the increase in the P62 level is there. That might be one of the reason of that supported reason. So this is that P62 molecule, and we can find from this P62 molecule that the P62 basically carries the protein that is destined to be degraded to the autophagosome, and in the autophagosome it got degraded once with the lysosome got fused with that autophagosomes. Along with that adapter protein that is carrying, it is basically an adapter of the cargo molecule that is carrying the molecules that is destined to be degraded to the autophagosome where it needs to be degraded. So this P62 level should be decreased if the autophagy is completed, but we find it to be increased. So there might be some of the process in the autophagic flux that might be stopped or halted somewhere in the autophagic flux. So this P62 protein has three important domains. The many domains, out of them I am interested with these three: LC3B binding domain. This is the domain which binds with the autophagosome marker protein LC3B. This is a KEAP1 binding domain. This KEAP1 is the important protein, and this the ubiquitinated proteins are being attached with it. So from here we find that also that the P62 from these two literatures study, we find that P62 also interacts with the RPT1. RPT1 is one of the important protein of the chaperone system, and we find that in the 0 days, in 8 days post-infection, there is a considerable increase in the interaction of the P62 with RPT1, which might be a result of the increase in the proteasomal degradation level. We have assayed the proteasomal degradation assay by trypsin-like, caspase-like, and chymotrypsin-like. All the 3 modes of proteasomal degradation increases. There is an increase in the ubiquitin level as well by the immunohistochemical staining and also by the Western blotting. So we have also checked the Okay, so we also studied the phosphorylation status of the P62 protein. So in the phosphorylation status, we find that the 2 types of phosphorylation, like serine 403 phosphorylation level and serine 349 positions phosphorylation level increases. This 349 position phosphorylation level increase actually indicates that the P62 keep-on interaction is increasing with the days post-infection, as with the increase in the days post-infection. And this NRF2 and keep-on basically remain with each other, interacted with each other, and this keep-on, when freed up from the NRF2, this NRF2 can go into the cytoplasm and regulate a multiple number of genes. So in our cases, we find that the P62 and keep-on interaction increases with the immunoprecipitation, which means that the P62 in the cytoplasm remains interacted with the keep-on, and the NRF2 molecule interacts with the NRF2 molecules fails to remain in touch with the keep-on. Hence, this NRF2 molecule is now freed up, and it is now free to move to the nucleus and regulate the gene expression of certain genes. So interestingly, we find that this P62, which we find that to interact with keep-on and this interaction increases with the days post-infection of the malarial mice in the splenocytes. This NRF2 then gets freed up and NRF2 translocated to the nucleus, and this NRF2 sits on the promoter region of the P62 gene itself. That is one of the most important, interesting thing, that the P62 gene itself, the NRF2 binds to the promoter and causes the gene expression or the increases in the expression of the P62 level, which ultimately forms in one loop. So this is a loop that is going on, and that might be a cause that this P62 level. We find that initial part of our study, we find the level of the P62 to be increased. That might be one of the reasons for that. So in a nutshell, this is my study, the proteasomal degradation, the 3 modes of and the autophagic process, and this P62 plays the important role, and P62 interacts with the RPT1 to initiate the proteasomal degradation. It interacts with LC3B so that autophagy can happen. It also interacts with the keep-on so that the NRF2 translocated into the nucleus. And this is the future of my study, that we want to go into the NF-kappa beta pathway, because every data that we have right now is linking to that NF-kappa beta pathway, and it might be one of the key factors that might be controlling the malarial infection. So this is my supervisor. This is my department of zoology, University of Calcutta. I want to thank my supervisor, my fellowship organization, the UGC, the project funded by DBT of India, BRNS, and this is the lab group. This is Torun Keswani, this is Shamrat Sarkar and Shoikat Mukherjee on the malarial biology, and this is our whole group. And this is my University of Calcutta, my Science College campus. Thank you for thanks. I want to also give thanks to the GRDS organizers for giving me the opportunity to speak over here, and I want to thanks to all of you for your presence. Thank you.
Any questions? Anything you want to ask? How the patient community will be benefited by it?
Okay, so the thing is that the patients, so we are right now working on the mice model, but we are actually, my main focus is that when the pathogen is inserted by a cell, by a phagocytosis mechanism, it might be that the pathogen might not be completely degraded, and if it is not degraded over in the cells, it cannot be presented by MHC class 2 and the other immune cells are not being triggered. to fight against the infection. That might be a reason that that the infection doesn't causes so many deaths. So that's why we are trying to figure out that whether if we use an autophagic inducer, so that if we find that autophagic block is there in some point, so we are trying to first figure out that at what points the autophagy is blocked, and now we are trying to use some inducers so that those blocks can be reversed. And if that autophagic process can be completed, probably according to the other references that we have, that might be play a key role in the combating the diseases in future. Thank you.